Good morning again. My name is Georg Eichler. I used to be a clinical pharmacologist in academia. I'm now a regulator. For 5 years I've been the senior medical officer of the European Union. And I'm also very closely involved in a group at the MIT called NUDICS that look into different ways of licensing and drug development procedures. They're called NUDICS. So this is very close to my heart. Now, this is called a cross-sector perspectives panel. Whatever it means, I'm proud to announce that you have 3 excellent speakers on the agenda, but before you can hear them, you have to put up with me. That is called an exercise in delayed gratification. So, I will be echoing some, but hopefully not too much, of what Dr. Cox from FDA had said, and I think it's a good thing if I echo it. It would be worse if I disagreed with him. So, let's talk about new paradigms for TB drug development. Just a few thoughts from myself. And what I want to do over the couple of next minutes is remind all of us about the basics of research. And then I will remind you that regulators have to wear 2 hats. That is, if they are reasonably smart. One hat is that of a gatekeeper. Basically, that is someone who says no. And the other is that of an enabler. That is someone who should say, yeah, yes, maybe. So let's start with Research 101. Let's pretend, forget TB for a moment. Let's pretend we're a bunch of scientists in the mid-20th century. We have discovered a new enzyme, and now we want to characterize that. And we know there is a number of variables that will impact on how that enzyme behaves. One, of course, is temperature. The other is concentration of substrate, concentration of inhibitor, and so on. There are many factors in the environment. Now, we have all learned that we do this by way of experiment. And an experiment in all of science, not just medicine, means that all factors except one are kept constant. So we first look at what is the influence of temperature, and during that period we will keep the concentration of substrate constant, of course. And then we go through step by step. Otherwise, if we modify all the variables at the same time, we will get some results, but we will not be able to interpret them. That is the basics of research. And that is also the basics of clinical drug development. So now I'm seeing this, and you'll recognize this. You have this picture in your pack, and this is what came from CPTR. And it says, interestingly, there are the steps at the top. This is how we would do our experiment with the enzyme again. This is how we would develop a drug today. You take one step with the left foot, then another step with the right foot. Try to do both at the same time. Well, that is exactly what we are hearing that we should be doing in future. And I'm joking, of course. I understand the imperative. But I want to point out the difficulties that come with that. So, what we are having is simply, and this is now the regulator as a gatekeeper, look, guys, you want me to tackle just too many variables at the same time. And they're all unknown. Let's go through these. We heard, both from Carl and from Dr. Cox, that we have to look into surrogate endpoints because the clinical endpoints, say 18, 24 months, relapse-free cure, is just too long away. It's just too difficult, too far out. So we need a surrogate endpoint. And we heard that there are some surrogate endpoints that look very promising. Have they been proven or validated, whatever word you want to choose? Well, I'm not sure. I would say this is one variable in the equation. Then we are hearing that we cannot afford the time because there is a public health crisis looming. We cannot go on through 15, 20 years of drug development. Therefore, and for another reason, because we want to contain resistance development, we should not look at individual drugs. We should consider the unit of development is not a drug. The unit of development is a regimen. So we will be looking at what's called in our slang new combos. Well, all very good, but that introduces another variable. What is the contribution of partner A versus partner B in that combo? And then we are looking at treatment shortening potential. I do understand that the 6-month treatment period is difficult in terms of patient retention and a number of other logistic issues out there in the field, but introducing shorter treatment courses introduces another variable, an unknown one. And then we're looking at different populations. You know those better than I do, and you understand those abbreviations better than I do. But of course, can we extrapolate from population A to population B? I have seen development programs that look at them in a sort of mixed scenario. It's unknown. And then, what is the optimal dosage in these combinations? So should we start with partner A in a combination and then follow on to partner B? Should they be started simultaneously? Should that be first line, the next one second line, etc.? All unknown. And then here's a big one. It only came up briefly in the discussion with Dr. Cox. Well, a drug is out the door. What now? And it is always said regulators don't regulate the practice of medicine. That's all very good. But sometimes, unfortunately, the practice of medicine has been regulating us. And that happened when we let out a drug, it went out, it was prescribed and utilized, shall we say, in a less than optimal way. And then we had to take it off the market again. We have a number of examples, not in the TB sector, but it happened in a range of therapeutic areas. So it isn't that simple, and we cannot just turn a blind eye to what will happen after that. So these are some of the unknown variables that I could think of. I trust people in the field who are more experts than I am could identify even more. So that leaves me with a situation like this. You're telling me, could you please solve this equation for me? And by the way, all the variables are unknown. Well, good luck. So the regulator in me that is a gatekeeper says, sorry, it can't be done. Too many variables. Let's go back to our tried and tested stepwise approach. Now, I do accept that this would be the purist school, but it would be a bit disingenuous. So can we try and also look at the enabling regulator's role? And for that, I say we have to go back and revisit the traditional paradigm of drug development and drug licensing. I said earlier I'm a clinical pharmacologist, and one of our heroes in the past is a guy called Lou Scheiner, who said drug development is always circles of learning and confirming. You first try to understand what your drug does, that's the learning phase, then you confirm that, you test your hypothesis, and then you see your friendly regulator and try to get a license. So that's where we are in the normal paradigm. What you people are asking us now is to basically turn that paradigm on its head. And now it's learn, then license, and then confirm. Because otherwise we will not do the trick. Otherwise we cannot come down from the 15 years to the 5 years. So something has to give. We will have to look at what kind of license that is. Is that an ordinary license, or could that be a conditional license, or whatever? But I think one thing is very clear. For those of you who grew up in the old paradigm, which is develop a drug, get a license, and then hand it over to your marketing department, and that was it. Please kiss that idea goodbye. What you want is a life cycle approach to research and licensing. So it doesn't end with a license. Now, having said that, here is my nasty little table of unknown variables. And what I want to do now is fill in those cells on the right-hand side, which could be potential solutions, or how can we approach this? So we said we have an endpoint that is promising, but it isn't yet validated or proven or whatever you want to call it. Well, what we like to see is a biomarker qualification program, and I understand that is underway. Could that be run in parallel with drug trials? Probably yes, but be aware that we're all taking a risk here together. But it's doable, certainly. The unit of development being a regimen. Now, this is a tricky one. As I said, the old regulatory thinking is if you have a combination, you should understand the contribution to efficacy and safety of each of the partners. Now, that is intelligent in a way, but I'm not entirely sure it's a priority. Imagine you are a patient. You are being given a combination, and it does you good. You have no adverse effects, it's effective. Are you interested to know how much of that efficacy came from A or B? No, it's your least concern. You couldn't care less. So I think this will depend on how it pans out. If you had in an ideal world a perfectly efficacious and perfectly safe regimen, do we need to care about the components? No. But if there was an issue with safety, if there was an issue with suboptimal dosing perhaps, then we will need to drill down and further identify how the combination partners play together. Then there is this idea of the treatment shortening potential. Of course this is valuable, but you need to assure us that you can follow up long term, so that maybe you can build that question into your development program and see, are we not worse than we would have been based on historical data or other comparators? What about the optimal, so yeah, what about the populations? We understand that there are several different subpopulations, and it is a bit disingenuous to say we will work them off one by one or in parallel. Can you learn from one to the other? Yes, probably you can. So to some extent you will probably have to do parallel RCTs in different populations, but to some extent you may be able to extrapolate. You will get those people onto the registry and maybe learn that way. And then there is this idea of optimal dosage, combination. I think my colleague from FDA said there are so many possible combinations and how you stagger them in time, etc., in dose. There is no way we can learn all that in the pre-licensing phase. So we will have to heavily rely on post-licensing observation. And then this last, my favorite hobby horse, because we were so burnt by this. Well, will you then just say, okay, here's the drug for anybody. It's a combination of 3 components. There are 3 different pills. Anyone can have as they please dose as you want. And if someone, even if it was developed as a combination, if someone wants to prescribe only the individual, well, that might not reassure us. So, here is my last slide. And trying to be an enabling regulator, not just a gatekeeper, I would say, well, maybe it can be done. But to help me, to help you. So I would like to see, first, a complete development plan. And when I say development plan, I don't mean what you call in the US the end of phase 2 meeting. That's not a development plan. That is a clinical trial plan for a phase 3 study. I want to see a complete development plan that goes far beyond the licensing, that includes all the post-licensing step and the learning and the confirming post-licensing, and maybe a second round of licensing. Show me a biomarker, or in that case it's an endpoint, it's a surrogate endpoint qualification plan. I think that is probably the easiest because it's already partly being done. I would say show me that the infrastructure is in place to monitor post-licensing treatment experience. Because even if you give me the greatest plan, that's my first bullet, but the infrastructure is not credibly in place, and you're doing these studies and the rollout in places where there is not a chance in hell that you could observe, then it's not reassuring. And then I would say, show me that the infrastructure is in place to monitor post-licensing drug utilization. So what will happen with those? Will we learn, or will these tablets or packs simply disappear somewhere into nirvana? And that in itself is also not enough, because monitoring is one thing. But show me also that you have infrastructure in place by way of restricted distribution schemes or whatever the case may be that will also manage the post-licensing drug utilization so that we don't fall flat on our face because we have been trying to take too many steps at the same time. So let's try to be an enabling regulator, but as I say, this is a partnership, and you will have to help us to help you. And I will stop with that, and thank you very much for your attention.